Good evening. 안녕하십니까? 한 캐나다 오픈 세미나에 참가해주신 모든 분들 환영합니다. 저는 오늘 행사 사회자를 맡은 김예언입니다. 반갑습니다. My name is Jennifer Kim and I'll be your MC for tonight. 주 토론토 총영사관은 포스트 코로나 시대, 다양한 분야의 협력을 활성화하기 위해 캐나다 한인 교수협의회와 공동으로 한 캐나다 오픈 세미나 시리즈를 진행하고 있습니다. 오늘 세미나는 바이러스 질병 분야 세계적 석학이신 강치룡 박사님께서 코로나19 백신 개발 전략 및 집단 면역 형성 가능성을 주제로 강연을 하십니다. The Korean Consulate in Toronto has organized an open seminar series in collaboration with the Korean Canadian University Professors Forum to promote cooperation in various fields for the post-COVID era. We'll be hearing from Dr. Chil-Yong Kang on COVID-19 vaccine development strategies and possibility of establishing a herd immunity against COVID-19. Dr. Kang is a world-class virologist and professor of virology in the Department of Microbiology and Immunology, Schulich School of Medicine and Dentistry at the University of Western Ontario in Canada. 강치룡 박사님은 현재 웨스턴 온타리오대 슐릭 의과 및 치과대학 미생물 및 면역학과 교수이자 분자 바이러스학 전문가로 활동하고 계십니다. 강 박사님께서는 코로나19 백신 연구뿐만 아니라 최첨단 유전공학 및 생명공학 기술을 활용하여 B형 바이러스, C형 간염, 에볼라 바이러스, 메르스, 지카 바이러스 소두증, HIV 에이즈 등 다양한 인체 특이 바이러스에 대한 항바이러스 치료제 및 백신 개발 등 다양한 연구를 진행 중이시며 300여 편의 논문을 발표하고 70여 개국에서 15개의 국제 특허를 보유하고 계십니다. 이러한 공로를 인정받아 한 캐나다 헤리티지 문화상 제35호 과학의 날 과학기술 훈장 혁신장, 캐나다 총독 엘리자베스 여왕 2세 다이아몬드 주빌리 메달, 한국과학기술단체 총연합회 올해의 과학자상, 온타리오주 혁신센터상을 수상하셨으며 현재 캐나다 왕립학술원과 대한민국 과학기술한림원 평생회원으로 활동하고 계십니다. Dr. Kang's research in molecular virology includes the development of viral-specific antiviral therapeutic agents and vaccines against various human viral diseases, including hepatitis C, Ebola hemorrhagic fever, MERS, Zika virus microcephaly, and HIV and AIDS. Dr. Kang has published over 300 scientific papers in fields of virology, medicine, immunology, and molecular genetics. He holds 15 international biotechnology patents in over 70 countries. 오늘 강 박사님과 세미나를 통해 코로나19 백신에 관해 좀 더 알아가는 유익한 시간이 되기를 바랍니다. 강연 후에는 질의응답 시간도 마련되어 있으니 오늘 강연 주제에 대해 궁금하신 점은 채팅창을 활용해 질문을 남겨주시기 바랍니다. 그럼 이제 강치룡 박사님을 모시겠습니다. Please welcome Dr. Kang.
Today, I'd like to introduce all the work that we do, but first, I prepared slides in such a way that the non-specialists can understand what we are doing. Now, as of yesterday, the whole world has total number of 172 million cases, confirmed cases of COVID-19, and with the 3.7 million deaths, and that accounts for approximately 2.1% mortality. And in Canada, we have 1.38 million cases with 25,000 deaths. And Korea, remarkably, has done extremely well in preventing this COVID-19 pandemic. And although Korea has more than 50 million people in a population in South Korea, and it had only 142,000 cases with little less than 2,000 deaths. And this was remarkable. So This is the world trend. Now the vaccination is going on, and we like to learn something about the vaccine itself. Now, how do we prepare the COVID-19 vaccine to prevent the virus infections? And there are many different strategies that are being used to develop the vaccines against the COVID-19. And first of all, let's just discuss what kind of commercially available vaccines there are to prevent many different types of viral diseases. There are four types of commercially available vaccines, and to prevent the many different types of viral infections. First type is attenuated live virus vaccines. And this attenuated live virus vaccine represents the virus which is non-pathogenic, but it can trigger immune responses to generate neutralizing antibodies. The types of the vaccines that we are using, this attenuated live virus vaccine is oral polio, measles, mumps, rubella, yellow fever, smallpox, varicella zoster, and the many other viruses vaccines, including animal vaccines. The second type of the vaccine is inactivated, kill the whole virus vaccine. So what you can do is purify the virus and inactivate them and use that as immunogen to trigger immune responses. And examples of that inactivated virus vaccines are injectable poliovirus vaccines, rabies virus vaccines, hepatitis A, influenza, Japanese encephalitis, and the many more animal vaccines are used this strategy. And the next two different types of vaccines, first of all, recombinant subunit vaccine is a relatively new strategy that people can clone the DNA of the genes and then have it expressed in the animal cells or eukaryotic cells. I'll explain this more details later. And make proteins, and those proteins are used as a source of vaccines. The examples of that is hepatitis B virus vaccines. I think many of you have had that. And also HPV vaccines. These are human papillomavirus vaccines. This is against the cervical carcinomas. And also there is another type of vaccine, which is a vector-based vaccine. You can use the vector, another type of virus which doesn't cause any disease in human, but you can insert the gene of interest into this non-disease causing virus and use that as a vaccine. And one example is recombinant Ebola virus vaccines. So let's just look at what type of blood cells we have in our body, which will trigger immune responses to make us to be resistant to virus infection. We have bone marrow. So we have a bone marrow which generates the stem cells. That's a pluripotent stem cells are produced from bone marrow, and that stem cell is differentiated into many different types of the blood cells. Now, blood cells that are important for the immune responses are so-called B cells and T cells. Specifically, the helper T cells helps the B cells to be mature and produce antibodies. Now, what happens is that the B cell recognize the antigen, such as, let's say, hepatitis B virus or SARS-CoV-2 antigen spike proteins, and then it can be activated by helper T cells through so-called MHC class 2, is major histocompatibility antigen. And these helper T cells carry the so-called cytokine known as the CD4. So they interact. B cells interact with the T cells, and then this B cell be actually activated. And in a way, the simple way of saying is that it can be educated And these educated B cells become a plasma cell, and that will produce antibodies. These antibodies then interact with the antigen that has been initially associated with the B cells. Now, another subset of B cells become so-called memory cells. These memory cells are important to have the boost immunizations. So when you have the prime immunization with the first vaccine, and then you go in with the second time 3 or 4 weeks later with the booster vaccination, these memory cells will produce much faster antibodies so that you can be protected from the virus infections. So these are the immune system in a very simple way to explain. And then next, when we have the viruses such as the SARS-CoV-2, which recognizes on the cell surface a receptor is known as ACE2. It's called angiotensin converting enzyme 2, which is a receptor for this SARS-CoV-2, which causes COVID-19. And this spike protein interacts with receptor and gets into the cell and causes replication and diseases. Now, if you make antibody by vaccination, these antibodies can cover up or mask these spike proteins so that these spike proteins cannot interact with the receptor ACE2 so that the virus cannot get into the cells to establish infections. This is what we want. So vaccine, in a sense, is to try to make these antibodies to cover up these virus particles before they enter our cells. That's the whole strategy. Now let's look at what the SARS-CoV-2 virus look like. These are the spherical particles, and that is about 130 nanometers in diameter. It's a tiny and very small virus particles consist of the RNA as a genome, and that RNA is a positive sense RNA genome containing 30,000 nucleotides. Our body consists of 3.3 billion base pairs of the nucleotides, and this virus has only 30,000 nucleotides. However, the infection of this virus can cause diseases, okay? And this virus actually produces the 4 different structural proteins: the envelope protein, the membrane protein, and nucleocapsid protein, and spike protein. All the vaccine developers are using this spike protein as a target antigen to make antibody against this spike protein. Spike protein exists as a trimer. It's 3 molecules together stick out from the virus particles. And that spike protein recognizes the receptor ACE2 and to infect the cells. So everybody who are making the vaccine against the SARS-CoV-2 are using the spike protein gene. Now, as I told you, that this is RNA virus. In order to manipulate RNA in a way, you have to convert this RNA to a DNA by enzyme called reverse transcriptase. So we can convert RNA into DNA, and then we can manipulate the genes the way that we want. Now, there are 7 different types of the COVID-19 vaccines being developed. Over 180 COVID-19 vaccines are being developed in the world right now. And these include, as I mentioned before, inactivated virus vaccines, weakened virus, which is the attenuated virus vaccines, the viral vector-based vaccine with a replicating virus vector and non-replicating virus vector, and nucleic acid vaccines is DNA virus vaccines and RNA vaccines. Now, and then there are protein-based vaccines, which includes subunits of proteins as well as virus-like particles. And this is the common strategy to develop SARS-CoV-2 vaccine. However, the RNA vaccine is now being used, and non-replicating the vector-based vaccine is now available to prevent infections. Now let's go through each one of these strategies to understand how these different strategies work. First of all, inactivated virus vaccines, we can purify the SARS-CoV-2. This is a virus particle. And these purified virus particles consist of these spike proteins. And these spike proteins will trigger immune responses. So one can produce large quantities of this virus and purify them and inactivate them. You can inactivate them by chemical or radiation. And then you can use this kill the whole virus as a vaccine source. And these kill the whole virus needs so-called adjuvant. Adjuvant is immunostimulant to produce more efficient immune responses. So you mix these purified virus particles with adjuvant and make vaccines. Now, companies who are making these kind of vaccines are in China, for example, Sinopharm, they showed 78% efficacy, and the Sinovac China and had the clinical trials in Turkey and Brazil, and they showed over 80% efficacy. And the Bharat Biotech of India, they showed also 81% efficacy. And there are other companies who are making this kill the whole virus vaccines. Okay. The next strategy is the live attenuated virus vaccines. Because it is difficult to produce the live attenuated virus strain, I mean, it takes a long time to characterize the attenuated strain of SARS-CoV-2. Therefore, this strategy is being used, however, not very popular because it takes a long time to develop vaccines. So what happens if you have a vaccine is that you can have this live virus and vaccinate them. Vaccinated individuals will have this live virus infecting, and this live virus will produce messenger RNAs, and this messenger RNA will produce proteins. It's called translation. So this virus-infected cells, will produce the live virus as well as protein. And these can be used to trigger immune responses. But as I said, because it takes a long time, so people are not using strategy very much. The next strategy is spike protein-based vaccine. It's a protein-based vaccine. You can express spike proteins using cloned DNA of spike protein gene, and you can express them in eukaryotic cells, and then purified virus protein can be then combined with adjuvant, which is immunostimulant, and trigger immune responses. This spike protein vaccine is being developed by Novavax, Sanofi GSK, and the Vaccine Institute of China, and the many other companies and institutes are using this strategy. And this strategy have been used for hepatitis B virus vaccines and the human papillomavirus vaccines, and this strategy works. But it took longer than expected, so that I think the Novavax is just about to launch their or get the license for the usage of these vaccines. And I understand that Korea has the contract with the Novavax to manufacture this vaccine in Korea for the distributions. Next strategy is the DNA vaccines. The spike protein gene can be cloned into a plasmid DNA, and that DNA can be used as a source of genes of spike protein, so that recombinant DNA can be transfected into the vaccinees or the person, and the person who gets this DNA will see that the DNA gets into the nucleus of the virus or transfected cells, and this DNA will be transcribed to make a messenger RNA. This messenger RNA then leaves the nucleus, get into the cytoplasm, and translate this messenger RNA to make proteins, and that protein will be secreted out of the cells to trigger immune responses. This whole process is, again, highly inefficient. Therefore, the companies who develop this kind of DNA vaccine have tried this, but their efficacy was relatively low. So I don't know whether they are continuing or not, but there are other companies, especially the companies who are collaborating with US and Korea, and also there's a Japanese company, and the Korean-owned company is producing this DNA vaccine, but this is not being used so far for the prevention of the SARS-CoV-2. Next vaccine is brand new technology. This is known as mRNA vaccine. This is instead of using the genome of spike protein, one synthesizes this RNA, which can be put into the cells to be translated by putting this RNA into a tiny lipid particles. It's a nanoparticle. Lipids can carry this RNA, messenger RNA, into the cells, and these RNAs will be translated to make a spike protein, and these proteins will be secreted from these transfected cells, or RNA enter the cells, and these will produce spike proteins to trigger immune responses. Initially, there was some skepticism about this technology because in the cells, you can have a loss of enzyme which is known as RNase, ribonuclease, which will chew up these,
it's 19 hours.
mRNAs. But it turns out that this strategy worked out well. And now you know that Pfizer-BioNTech showed that 95% efficacy with mRNA vaccine, and Moderna showed 94% efficacy with these mRNA vaccines. And there are other companies are producing these mRNA vaccines. The next strategy is replication incompetent or replication defective recombinant adenoviruses. That is, you can use the cold virus known as adenovirus. Adenoviruses can cause cold in chimpanzees, humans, and gorilla, and so on. And these adenoviruses, you can make it such a way that this virus doesn't cause any cold, and also is safe. And you can insert SARS-CoV-2 spike protein gene into this adenovirus. By the way, this is a DNA virus. That you spike protein gene into DNA. That DNA can be inserted into this adenovirus and make it recombinant adenovirus. These recombinant adenoviruses are defective in replication because they delete one gene known as E1A gene, and E1A gene is necessary for virus production. But you delete that. Therefore, this virus can infect the cells, but they cannot reproduce. These recombinant adenoviruses carry the genetic code which can produce the spike protein messenger RNA. That messenger RNA can be translated to make spike proteins, and that will release into the media or extracellular portion, and these spike proteins can then trigger immune responses. And that this process is relatively efficient, but they do not produce more the adenoviruses. Therefore, they have to overload these viruses. So they are putting as many as 50 billion virus particles per dose of these vaccines, okay? And now the companies who are making this recombinant adenovirus gene or vaccines are Oxford AstraZeneca. This is developed in Oxford University in collaboration with AstraZeneca. And this is a Sputnik, which is a Russian vaccine. And Oxford uses chimpanzee adenovirus. Russian group, Gamaleya Institute, uses a human adenovirus 5 and adenovirus 26. The reason that they're using two different types of adenoviruses is because that one can make vector immunity against adenovirus 5. So if you go in with same adenovirus, it may prevent the boost effect. Therefore, they're using two different adenoviruses, and that has a better boost effect. And therefore, they show you the 91% efficacy, whereas the chimpanzee adenovirus, they use the same virus for the prime boost, and they showed about 80% efficacy. And there is a Johnson & Johnson vaccine using adenovirus 26, and they show only 66% because they're using single immunization or single dose vaccines. Because if you go in with the second time with adenovirus 26 vaccine, that your immunity against adenovirus, which is a vector immunity, will prevent the virus infection. Therefore, you won't have a much strong boost effects. And there are other companies that are using human adenovirus 5 or gorilla adenovirus 5. This is from Italy. So they show the good immune responses with the gorilla adenoviruses. So this is the vector-based vaccine, and we are using the vaccine here in Canada and also in Korea. And the Russians have offered their Sputnik adenovirus-based vaccines, and Johnson & Johnson is going to distribute their vaccines soon. Finally, this is the replication competent recombinant virus vaccine. We are using this, and this is our vaccine. We are using this strategy using Western Sumogen recombinant VSV COVID vaccine. And this one is replication competent. So when we insert the genes into this VSV, vesicular stomatitis virus, and the genes that is in this the virus will be expressed to produce proteins, and this will be released into the media, and that will trigger immune responses. Not only the soluble proteins of the spike protein, but also one can have the recombinant VSV, which we know as a pseudovirions, which consists of the two different glycoproteins. One is VSV-G protein, and also the coronavirus, which is spike protein. So it's a combined. This virus particle is much more immunogenic, and it does not require adjuvant to immune response. So these are the different strategies that people are using for the vaccine development. I like to take about 5 or 6 minutes to introduce what we have done with our vector system. We are using VSV, vesicular stomatitis virus, as a vector system to produce our vaccine. And we have developed this platform technology for the production of the vaccines against Ebola, Zika, hepatitis C, and HIV. And so we are using this for now SARS-CoV-2 vaccines. We have generated avirulent M gene mutants so that the virus itself does not kill cells, it doesn't have side effects. And these bullet-shaped particles can carry the gene of interest, in this case, SARS-CoV-2 gene. So like this spike protein gene, which consists of the 3,822 nucleotides, this is a genome length, and we take this gene into the VSV. So we are taking the spike protein gene by itself, or we modify the spike protein gene in such a way that we can put the spike proteins onto the VSV pseudovirions. So we are putting so-called GTC, is a VSV glycoprotein transmembrane domain and cytoplasmic tail. And if we add this onto this full-length spike protein, it can be associated with the virus particle. That's the other. So we have a system known as reverse genetics, we can recover these recombinant viruses by reverse genetics. I won't go into the details of it. Later on, if anyone wants to know more details about it, I can explain that later. So we can recover the virus particles by this so-called VSB reverse genetics. And once we recover that, then we can look at the purified virus particles. Now, this is the pseudovirions, consist of not only the VSV proteins, these are the VSV proteins here, and they consist of the full-length spike protein and S1 subunit. This is the done. We know we are using this as the Western blot using antibody against RBD, so receptor binding domain, which can recognize the S1 subunit, and anti-S2, which will recognize S2 subunits. So we have the full length and S1 and S2 are produced and incorporated into these pseudovirions. So we are using this as a vaccine to prevent SARS-CoV-2 infection. And we have done the immune response studies, and I'll just explain that this produces enormous amount of the antibodies. This is the log of reciprocal of the IgG, which is immunoglobulin G, endpoint titers. We can get almost the 1 million, one in 1 million dilutions can show the good positive response. And then we also show that the modified gene of spike protein can produce very, very strong neutralizing antibodies. By the way, the titer of the neutralizing antibodies, comparing with any other system, we have at least 10 times more neutralizing antibodies produced, which means that it can prevent all the SARS-CoV-2 infections. Then we do the challenge studies. We can do the challenge studies using transgenic mouse. That mouse are normally not acceptable for SARS-CoV-2 infection. However, you can insert the human ACE2 gene into the mouse so that the mouse can become susceptible for SARS-CoV-2 infection. Those are known as ACE2 transgenic mouse, and we can then infect those with the live virus of SARS-CoV-2. Vaccinated animals all show complete protection, but the control animals show the virus production in the lung. So it is completely protectable. So this vaccine is now ready to produce. So what's next? We are now producing this vaccine in a CMO. CMO is a contracted manufacturing organization. And then we are also doing the GLP testing simultaneously. And GLP is a good laboratory practice testing for the vaccine safety and immunogenicity. And then we will go into the phase 1, phase 2 human clinical trials in Korea, Canada, or in the US. And we'll do the phase 3 clinical trials in the actually highly prevalent countries such as India or Brazil or Canada. And so we can test that, and then we'll have our vaccine ready for the worldwide distribution. So that's where we are now. So now let's talk about the herd immunity. Now, herd immunity means that if you vaccinate large number of or high percentage of the people, let's say 75% of the people, then virus has no place to go, so that the virus will disappear. So is it possible to establish a herd immunity against SARS-CoV-2? Now, we need to vaccinate about 5.8 billion people in the world in order to establish effective herd immunity, because we have currently 7.8 billion population on this earth. And 75% of the 7.8 billion is about 5.8 billion. So how can we vaccinate 5.8 billion people around the world? Okay. Now, currently available vaccines, mRNA vaccine is Pfizer-BioNTech, 95% efficacy, Moderna, 94% efficacy, is very good. And then we have the AstraZeneca vaccines. We have this Russian vaccine. We have Johnson vaccine. Also, CanSino Biologics makes adenoviruses. In addition, there are some protein-based vaccines actually in the pipeline, and Novavax and Sanofi and many other companies are producing protein-based vaccines. Can we have enough vaccine to distribute to establish the herd immunity? Now let's look at the impact of the vaccination. You heard about India and US had a tremendous number of cases of the confirmed cases of the SARS-CoV-2 infections. Now, United States has now over 160 million vaccinations, and India is following right after that. Of course, India has a lot more population, 3.4 billion people in India, and US has about 380 million people. But as a result of this vaccination, this is the COVID-19 vaccination in Brazil, United Kingdom, Germany, France, Italy, and Canada. Canada is following now. And we have 38 million people. We have vaccinated about 56%, at least the first dose of the vaccines in Canada. So does this vaccination have any kind of impact on the daily cases of COVID-19? The answer is yes. You can see that the US had as many as 250,000 cases per day. This is the daily confirmed cases of COVID-19 in different countries. US had as many as 250,000 cases in the late last year and the beginning of this year. But then it falls dramatically fast after the vaccination. India has recorded almost 400,000 daily cases of COVID-19, and that case is now dropping down to 150,000 as a result of the vaccination. Other countries have followed the same way. Now, because of this vaccination, now we can see that dramatic decrease of the COVID-19 cases. Now, is it true that the world has only 172 million confirmed cases? Now, when you look at the cases identified in different countries, upper middle income countries and high income countries showed largest number of SARS-CoV-2 infections, not because it is highest in high income countries, because they have infrastructure to test people, more people, so that we know more people positive, the SARS-CoV-2 positive individuals in high income countries and upper middle income countries. But when you look at low income countries and lower middle income countries, the number of cases are very small. This may be misleading because we are not testing them enough. If you test them all, same as in high income countries, this 172 million cases may jump to maybe 500 million cases because we are not testing in these countries. So can we then establish herd immunity? Now then, it comes out with another problem. That is the variants. You heard about the variants of SARS-CoV-2. Okay, the variants are initially isolated from Nigeria, and that is the change of a single amino acid. Now, the SARS-CoV-2 spike protein has 1,273 amino acids. The protein, the building block of the protein is known as amino acid. Single letter code is recognizing one of the 20 different amino acids here. So that starting from methionine, finishing with the T here all the way, and we can show that there is a single amino acid changes here, and or sometimes deletions of the amino acids. Now, what I have highlighted here, the yellow portion of this spike protein is within this S1 subunit. This is S1 subunit, and this is S2 subunit. S1 subunit has the region which can interact with the ACE2 SARS-CoV-2, the receptor. And if you have a mutation in this, the interacting region of the spike protein, you may have a problem preventing the infection by vaccination with the currently available vaccines. Now, when you look at these mutants, like a UK mutant has one mutation in this yellow portion. This is the asparagine converting to the tyrosine in position 501. And then outside of this, the binding domain, there is another mutation which is called proline changing to the histidine. This is the portion here. And then there are two deletions here. This is a UK mutant, it's called B117. The Danish mutant has one mutation in this yellow portion, that is tyrosine changing to the phenylalanine, that is staying right here. Okay. And this South African variant is a very serious variant which has 3 mutations in this, the binding domain of spike protein. That has the asparagine changing to tyrosine right here. Oops. And the lysine changing to asparagine, which is K417 right here. And then there is the glutamine changing to the lysine, which is right here. So that these 3 changes may make a South African variant resistant to the commercially available vaccines from, let's say, AstraZeneca, Pfizer, or Moderna. So we have to see it because no one has done the clinical analysis of this, that we are doing those experiments right now. And Brazilian isolates, variants, consist of the 2 of the 3 of South African variant, that is N501Y and E484K. And in addition, US has the variant is called L452R, which is right here, again, within this binding domain. And now Indian variants consist of L52R, same as US variant, and in addition, they have other variants or changes in other portions. So what would happen if you use the currently available vaccine? Can we prevent all these variants or not? Is remain to be seen. And therefore, we are generating now vaccine against this important variant, which is the South African variant, and then the variant which can cover the Indian variants, so that one can use South African variant, Indian variant, or combination of all variants. So we made the South African variant, and this is now ready to be tested, and we are going to test in animals. They produce very good immune, the proteins, so that they will trigger good immune responses. So now the cumulated cases of this, the SARS-CoV-2, the confirmed cases, can we make this straight line so that there is no more cases, or will that increase with the time? And it depends on the efficacy of the vaccines, and also how much can we distribute these vaccines to developed or developing countries. Now, if we can distribute these vaccines to the low-income countries and the middle-income countries, we should be able to establish the herd immunity. Not until then. Therefore, World Health Organization has established so-called COVAX, which is the organization which will equitably distribute the vaccines throughout the world. And hopefully that COVAX program will work. Now, can we then see the drop of the daily confirmed COVID-19 cases down to baseline or zero. That remains to be seen, but looks like we can achieve that by vaccination. So I'm an optimist. I like to see that the COVID-19 pandemic will be terminated in a couple of years' time with the proactive vaccination. Now, I like to end with some positive notes. Now, I like to introduce two different diseases. Number one is the smallpox. Smallpox, in a history, that it has affected 900 million people, and 20th century alone, we lost 300 million people due to the smallpox infection. This is the patients with the smallpox infection, and patients who recovered from smallpox, they had the scars on the face. And we used to see many of these people. Now we don't see many more because smallpox has been eradicated. So I like to show you some positive notes on that. Smallpox vaccine has been developed by Dr. Edward Jenner in Britain, and he used in 1796 using cowpox virus to prevent smallpox infection. The cowpox virus is known as vaccinia virus. So the French scientist, Dr. Louis Pasteur, coined the name vaccine coming from vaccinia virus as a generic name for the substance which can prevent infection of infectious diseases. So today we are using vaccine, word vaccine, in actually honor of Jenner's discovery, which was proposed by Louis Pasteur. Okay, now you know why we are calling these preventive substances as vaccines. Now, we used to get the scratch of our arm, and with the cowpox, which is vaccinia virus, and we get the scars. And those of you who have been born before 1970, you must have those scars because we've been vaccinated. We all carry this scar on the arm. Now, because of this devastating infectious disease, WHO has launched the worldwide smallpox eradication program in 1967. And in 11 years' time, 1978, there was not a single case of smallpox reported in the world. This is complete eradication of devastating disease, infectious disease, due to the vaccination, okay? And this is remarkable, that this is the greatest achievement in infectious disease in human history. So the smallpox used to affect millions of people. Jenner introduced his vaccination in 1796, and WHO launched the smallpox eradication program in 1967. As of 1978, there is not a single case of smallpox in the world. Therefore, anybody who was born after 1978, you did not get the vaccination against the smallpox because it was not necessary. This is indeed a great achievement in infectious diseases. Next disease is a poliomyelitis. It's a poliovirus. And poliovirus is RNA virus. There are 3 different types. And this alone also infected many, many millions of people. And for example, in 1988, there were over 35 million infections with 350,000 paralytic cases. There's a 1% of the polio infection leads to the paralytic poliomyelitis. Thanks to the Dr. Jonas Salk and Albert Sabin, they produced 2 different types of vaccines. The Jonas Salk produced the Salk vaccine, which is the kill the whole virus vaccine, and the Albert Sabin produced live virus vaccine. And this disease is almost eradicated because the effective vaccination. This is the statistics from 1912 to 2001 in US, and introduction of the Salk vaccine and then Sabin's vaccine in 1961. As of today, there's very almost zero cases of the poliomyelitis in United States. This is, in fact, is another success story of the vaccination. So we can eliminate the devastating disease. Okay. So today, what we have done is we have discussed about the SARS-CoV-2 and the success stories of the smallpox eradication and the poliomyelitis eradication. And my laboratory is in Western University, and we get sponsorship from CIHR, it's the Canadian Institute of Health Research, Health Canada, and also National Research Council, and known as Sumogen, who sponsors our research. And I'd like to thank you, and I'll be happy to discuss any issues related to my presentations. Thank you.
네, 강연 중간중간에 몇 가지 질문이 들어왔기 때문에 그 질문들부터 제가 설명을 해드리겠습니다. The first question is, how can we know if the antibody has actually been made after 2 times of vaccination? 백신 접종 후에 우리 몸 안에 항체가 형성되었다는 것을 어떻게 알 수 있을까요?
We can collect the blood from vaccinated individuals, and we can check the level of antibodies in the blood. So that way we can see whether or not that individual has protective immunity.
And the follow-up question is, how long does it normally take for a regular person to actually develop antibody? We hear a number of days or a couple of weeks. In general, how long should we wait to think that we have antibody in our bodies?
Right. Our body reacts to the foreign substance, such as the virus. We can make antibodies against those foreign substances in about 2 weeks' time. And when you get the reintroduction of the same antigen or same virus, then you get the boost effect, so that you get the stronger immune responses. And therefore, we recommend that normally the first primary immunization, and you wait for about 3 weeks and then get the booster shot. And that will give you stronger immune responses, because our body consists of the immune system which can be trained to be more effective in producing more antibodies, so that the people can have stronger immune responses, so that they are much more protective against the virus infections.
Thank you. The next question we have is, what is your opinion on mixing vaccines? I know there's a lot of talk going on, and in fact, I think there may have been some announcement that Ontario may be looking into mixing vaccines. So what is your opinion on that? And also the waiting period between doses across different vaccines?
All right. So the mixing and mixing vaccines with 2 different sources is fine. For example, the mRNA vaccine produced by the Pfizer and also Moderna, and that can be used as the primary vaccines, and then you can have a booster vaccination with AstraZeneca vaccines. So it's okay to mix and match, because what you're doing is you're introducing antigen, which is actually spike protein, of the envelope protein of the SARS-CoV-2, so that we will make antibodies or react immune responses against that. So mix and match is okay, but we don't have enough data to show whether the same type of vaccine, in other words, you get the vaccination with the Pfizer vaccine, followed with the Pfizer vaccine boost immunizations, whether that will be as strong or even better than the Pfizer mixed with AstraZeneca. That has not been shown yet, but in terms of the introduction of antigens to our body, it is okay to mix and match. So you can have a vaccination with the one vaccine and then have a booster vaccination with another vaccine. That would be fine. So therefore, the government of Ontario, at least, is trying to encourage people to get 2 different vaccines. And now it takes approximately 3 weeks, or at least 2 weeks, to establish memory cells, and then so that you can go in with the booster vaccine for 3 weeks, and that would be fine. Now, because we don't have enough vaccines available to vaccinate everybody, let's say in Ontario, and therefore government is asking us to wait for maybe 2 or 3 months. But our memory cells will be there, and as soon as you go in with the booster vaccines, and that memory cell will wake up and produce more antibodies.
Okay, so it is okay for us to go after 3 weeks then?
Yes, yes.
Okay, that's good to hear. Thank you. So we have more questions coming in. So next question I have is, does 75% vaccination estimate for herd immunity assume a certain level of vaccine efficacy?
Yeah, it's 75% is not an accurate figure, but it's a suggested figure. If you have 75% of the people who are immune to the SARS-CoV-2 infection, then the 25% will be fine because the virus has no place to go. So if you have, it's known as a herd immunity. So we want to establish a herd immunity, that is a group immunity. And so if 75% of the people are immune to the SARS-CoV-2 infection, then the virus will have not so many places to infect, therefore they will disappear. And that's the whole idea.
Okay, thank you. The next question is, when a person gets immunized by a replication competent viral vector, will the person develop immune deference against the viral vector itself? If so, once the person receives a replication competent viral vector, the same viral vector can no longer be used for another antigen?
That's an excellent question. Yes. And you can have the replication competent viral vector, such as what we are using, the vesicular stomatitis virus. So that if you make the primary vaccine with, say, a vesicular stomatitis virus, one serotype, and then go in with the second serotype, which do not cross-react with the first serotype, you can have prime booster vaccination. And of course, you can have a vector immunity established so that you can have antibodies made against the vector itself, and that antibody can prevent the booster immunization if you use the same vector. But if you use the 2 different vectors, it'll be fine for the booster immunization. And so there is limitations for the multiple use of the same vector.
Thank you. 다음 질문은 제가 한국어로 드려보겠습니다. 현재 미국 같은 경우는 3차 접종 소식도 들려오고 또 코로나19 백신 예방주사처럼 매년 맞아야 할 것이다라는 그런 말들도 전해지고 있는데요. 박사님 생각 어떠신가요?
Do you want me to answer in Korean or in?
Yeah, you can do both.
Well, I'll do it in English because everybody can understand. The question itself is that if can we now, if I remember correctly, what you're saying is that do we need to be vaccinated every year?
Every year, or even a 3rd shot?
Yeah.
Do you think that will be necessary?
Actually, this virus infection will be different from flu virus infections because flu virus consists of the 8 different segments of the RNA, the genome. And then if you have the 2 different viruses infected one individual, you can have a shuffling of those genomes to create a new flu virus strain. It's known as reassortment. Therefore, the flu virus, the vaccine is being developed and produced every year because WHO recommends what kind of vaccine should be produced for the next flu season. Okay, and because it always produces different strains of the flu viruses. On the other hand, this COVID-19 or SARS-CoV-2 vaccines, it will not change as much as the flu virus does because it has a single genome length. It's of 30,000 nucleotides long, and you will have some mutations in it, but mainly the antibodies made against this, say, one virus, it should be effective for the other variants of the viruses. So that's remain to be seen, but not as problematic as a flu virus, because the flu virus shuffles, whereas this consists of single genome, and therefore we should be able to control it. And if we can establish 75% of the people and having antibodies, then the virus has no place to go, and therefore we can maybe terminate this the pandemic sometime later. It will take some time because in developed countries, it'll be fine because we can always diagnose who are infected. But in developing countries and the low-income countries, we don't know how many people are infected, and those people will spread the virus and much more efficiently. So that we need to go after them and diagnose them and see who needs more vaccinations. So I think it is very important to find out who are infected and who are not. And it's very difficult to do that in developing countries.
Thank you. The next question I have, I think it's a popular one. Risk of AstraZeneca compare with the risk of vaccines for other diseases?
The blood clots, well, this is a rare event. In fact, right now, they're talking about the 1 in 55,000 vaccinees, right? And it's a very rare event. And also, it depends on which vaccine you are using. And in fact, the AstraZeneca vaccine showed some blood clots. But when you look at the benefits compared to the risk that you're taking, there's a huge difference. So that even if there are some problems, I mean, 1 in 55,000 vaccinees show some problems, but the benefits that when you look at for the establishment of the herd immunity, I think we can tolerate that risk.
Thank you. All right, the next question I have is, what would be your thought on adverse effects on vaccines, including long-term effect?
We don't know about the long-term effects of the mRNA vaccine, which is produced by Moderna and Pfizer. And we don't know the long-term effects of the adenovirus-based vaccines, such as the AstraZeneca vaccines. And we don't know the long-term effects of the protein-based vaccines as yet. And so it remains to be seen, but we need to be immune against the virus infection. Therefore, we are taking very small risk to make sure that we are immune to the virus infections, right? And so there's always some degree of risk, but when you compare with the risk versus the benefits, benefit is much, much greater than the risk that we are taking. Therefore, all governments around the world are recommending the vaccination in order to establish this so-called herd immunity. The herd immunity is necessary to prevent this spread of this virus.
So there's a question regarding herd immunity. If 75% immunity is required for herd immunity, while vaccines are, let's say, 75% effective, does that mean that 100% vaccination would be necessary?
No, that's misleading. Actually, 75% of the people need to be vaccinated, okay? So that it's not 75% efficacy. 75% of the people, if you have 75 out of 100 people get vaccinated, then virus has no place to go because it's difficult to transmit this virus from one individual to the next individual if the 75% of the people are immune to the virus. So that's what the 75% comes in. Of course, if 100% people are immune to the vaccine, the virus, then it would be, the best scenario. But you cannot ask everybody to be vaccinated. And there are some groups who are rather reluctant to get the vaccination against any type of diseases.
The next question is, given how many variants there are going around right now, what are your opinions on the vaccines that we're currently getting? How effective are they against the variants?
Well, we remain to be determined. We have to see whether or not these currently available vaccines can prevent infection of all these variants. We actually, time will tell. And therefore, at the moment, for us, for example, if I talk about our research, we are making vaccine against different variants so that just in case the efficacy of the vaccine that currently available against these variants are low, then we can always use these variant-based vaccines. Right? That way, we can prevent the SARS-CoV-2 infections completely.
Thank you. You talked about side effects, especially around blood clots, but even just mild symptoms like tiredness or sore arms, etc. Some people are worried about those side effects, being too much after they get vaccinated. What are your thoughts, and how can we make sure that we're not too affected by it?
Yeah, well, we are taking. If the benefit is much greater than the risk that we are taking, then the human beings will take that benefit, right? So that vaccination, of course, when you vaccinate people get sore arms and sometimes some fever and all kind of small side effects. But when you look at the total benefits from the vaccination, you tolerate the risk that and also the vaccines, some people do experience with the side effects such as high fever and chill and sore arms for several days, not just a couple of days, depending on the vaccines that you're using. So for example, right now, mRNA vaccine produced by Moderna and the Pfizer, they show less complications compared to the adenovirus-based vaccines. But we'll see. But again, the benefit is much, much, much greater than the risk that you're taking. Therefore, we are taking vaccines.
Thank you. I know we're really focusing on vaccines tonight, but there is a question regarding masks. What are your thoughts on, when can we finally not have to wear masks? Okay,
if 75% of the population is immunized, then we don't need a mask, okay? Right now in Ontario, there are about 56%, if I'm not mistaken, the primary vaccinees. So that, and these are normally the elderly people. And if we can get the herd immunity meaning that everybody get vaccinated and the virus has no place to go, then we can take off our masks. Right now, there are two ways to think about wearing the mask. One is preventing the virus infection for yourself. That's the one. The other thing is that if you are not sure whether or not you are infected, especially young people, there are many asymptomatic infections. People do not show any symptoms, although they are positive with the coronavirus infections. So in that case, it is advisable to use N95 masks. Okay, now we are using masks to prevent two things. First of all, spread of your virus to the other people, or prevention, the virus coming from other people into your system. So that there are two ways. But I think we can take off our masks once we establish 75% people are vaccinated in Ontario, for example, or in the world. But it's going to be difficult to achieve that 75% vaccination around the world because there are developing countries who are not proactive in using the vaccine or vaccination. And only the developed countries, they really raise to vaccinate their own people first, right? And then the vaccine will be available later. And the WHO is trying to distribute the vaccines equitably using their system, but the vaccines are not produced enough to distribute 5.8 billion people around the world. So it will take time to get the herd immunity around the world.
Thank you. Next question is, can you say that COVID-19 is naturally caused or artificially caused? What do you think as a specialist in the field?
I assume that this question comes from, has anybody produced the COVID-19 or SARS-CoV-2 virus, right? Is that what you're asking?
Yeah, on purpose, I guess. Yeah.
We are actually, molecular biology has advanced so much in the last half century, right? But we cannot create the virus which can give the disease such as the COVID by creating the virus. I mean, some countries actually blame the China for production or generating the new virus, but it's not true. I don't believe in that. And so it's naturally causing, and the coronavirus is a very common virus among bats. And so this is naturally existing virus which somehow adapted to transmit the disease in humans.
Thank you. The next question is, when do you expect your vaccination be available for actual use?
Well, we are doing right now, manufacturing the vaccine for the phase 1-2 clinical trials. And hopefully that our vaccine will be available, hopefully, for the distribution in a year.
Okay. The next question I have is, if I have the natural antibody by any chance, do I still need the shot?
No, it's not necessary. You can check the level of antibodies against SARS-CoV-2, and it is possible to do that. And if you have, for example, unknown infection, so that you carry antibodies against SARS-CoV-2, and it depends on the level of antibodies. If you have a strong enough antibody level in your system, you don't need to be vaccinated. But not so many institutes or the laboratories test antibody levels for you. So it's easier to get vaccinated instead of testing yourself before you get vaccinated.
Got it. Thank you. So the next question I have is, thank you for the answers for the earlier question about potential artificiality of coronavirus. If so, why do some scientists do not rule out the possibility of accidental leaking?
Accidental leaking is different from creating the virus, okay? Now, Wuhan had the first case of this SARS-CoV-2, and that virus somehow spread so fast. So you can say it's accidental leaking because initially that institute or the city of Wuhan was trying to cover those patients, but then later on it was not possible to cover up. Remember that Wuhan had a lockdown, the entire city was locked down. However, the virus leaked out, then it's infecting the entire world. And so a lot of people have all kind of theories, but I don't believe in creating the virus which causes COVID-19.
Thank you. And I think this is my last question from the night. And I think it's just a clarification. When you talk about 75% of all people getting vaccinated for herd immunity, you mean fully vaccinated or just partially?
Well, I mean fully vaccinated. It's partial vaccination will give you a certain degree of protection. The degree of protection that we are talking about is severity of the disease. So if you carry certain level of antibodies against the SARS-CoV-2, then you can have a very mild symptoms if you get infection with the SARS-CoV-2. But if you are fully vaccinated and if your system produces very strong antibodies, then you can be completely protected.
Thank you. So this does wrap up our Q&A session. Once again, thank you, Dr. Kang, and thank you everyone for joining us tonight. We hope you enjoyed the session, and we hope you have a great night and stay safe. 감사합니다. 즐거운 저녁 되시고 다음 세미나 때 뵙도록 하겠습니다.
Thank you, Jennifer.